another biology video from A-Star Express. Today we're going to be talking about genetic modification. So what is genetic modification? It's when you change the genetic material of an organism by removing, changing or inserting individual genes. That's the IGCSE definition. So a gene is essentially the length of DNA or the length of bases that code for a particular protein. And so when we're going to change that for a particular organism by removing, changing, or inserting the genes, that is genetic modification. So today we're going to be seeing it for the example of insulin. So when we are using bacteria to produce human protein, so insulin, as we know, is used to regulate the blood sugar level. But when you have type 1 diabetes, etc., your body doesn't produce that hormone, that protein insulin anymore. So now pharmaceutical companies will produce insulin, and this is that process. So first, you in bacteria, the genetic material that we can use for this process is called plasmid, which is what I've drawn here, and it's like a circular form of DNA. So we take this plasmid and then we cut it. So the red lines are showing what the restriction enzymes do. So essentially, restriction enzymes will help us cut the plasmid and take out a section of the plasmid, leaving sticky ends. So what you see here, like these open ends, they'll have particular nitrogenous bases like adenine, thymine, or any other base. So then what we do is we take out the human gene. So here we have a human strand of DNA. So we can see this length of DNA is a particular gene. So let's say this is a human and this is the gene to code for the protein insulin. So what we'll do is we will take that part of the DNA out. We'll take that gene out. We'll isolate that gene again using the same restriction enzymes. And so we'll take that particular gene out, that gene that helps us code for insulin, leaving sticky ends here. So these sticky ends will be complementary to the sticky ends that are present in the plasmid. So what this means is the nitrogenous bases at these ends will be complementary to the nitrogenous bases on the plasmid, meaning that they can like bind to each other, and this section of DNA can bind to the plasmid. So that's what we have here. The human gene, because the sticky ends are complementary, they're going to bind here. And so what this is going to do is when this binds, this is going to form a recombinant plasmid. So this is done by DNA ligase. So what this does is it is going to like seal this plasmid with the human gene in it, forming a recombinant plasmid. And then this plasmid is going to be put back into the bacteria. These recombinant plasmids are going to be placed back into the bacteria. The bacteria are going to divide by binary fission. They're going to multiply, and then they can be put into a fermenter and they'll be grown, and then the protein insulin is going to be produced in larger numbers. And that is it for genetic modification. Thank you. Please request whatever video you like next in the comments below. Thank you.